Here we have some IB questions, some paper 1 multiple choice IB questions on the topic protein. So this is 2.4. So let's just get started. As usual, try these by yourself and I'll explain how you need to answer them afterwards. Okay, so which molecule diagram corresponds to the name? So here they have a name and then the way it should look like, the molecule. Okay, so the first one is saying D-ribose, and this molecule diagram is not D-ribose. This is glucose. This is a carbohydrate, glucose. A ribose only has 5 carbons. Okay, 5 carbons. But this molecule here has 6 carbons. 1, 2, 3, 4, 5, 6. And any structure like this that has 6 carbons is a glucose. Okay, so that's why A is not right. What about B? B, amino acid, and then they have this. This is right. We know all amino acids have the same basic structure. A nitrogen next to a carbon, which is next to a carbon, and then there's an R group on the central carbon. I'll show you a better picture here. Come down here. So right here, if we consider this to be a chain of amino acids, and we look at one amino acid, it has this basic structure, okay? The R group is a group that varies, okay? It's a group that can be any kind of structure, and we'll see later on in this video or the next video, okay? So this part here, the last carbon that has 2 oxygens and a hydrogen attached to it is the carboxyl group, and then we have this other group here called the amino group, okay, the NH2, the N and 2 H's, and then we have this R group, okay? That's the standard structure of an amino acid, okay? So basically, we know the answer is going to be B. Let's just eliminate these other ones. So C here is saying phospholipid, yet they are showing you a cholesterol structure. Okay, so again, maybe you don't know these. The reason why I chose this question is because you can do it if you know what an amino acid is, and that is part of this topic. So even if you didn't know any of A, C, or D, you would still be able to get this question right just because you should know what structure an amino acid has. So C is wrong because it's not a phospholipid. This is cholesterol. D is wrong because this is a ribose. Okay, notice 5 carbons. 1, 2, 3, 4, 5. Okay, so this is a ribose, not a glucose. So maybe A and D could have been swapped around. Okay, so the answer here is B. Okay, next one. Which is an effect of protein denaturation? So one of the key things we learn in this chapter, or this specific thing about protein, is that they can undergo a process called denaturation. Okay, let me show you what that means. So say we have, wait a second. Yeah, here. So say we have a full-on protein. So we know proteins are crazy 3-dimensional structures. Okay, they can be made up of alpha helixes and beta pleated sheets, which are all made up of course, amino acids that folded in some weird way. Now, denaturing is when you give some condition, such as a high temperature, a low pH, or anything like that, that destroys this 3D structure. Okay, so denaturing will break this 3D structure into its individual primary structures. Okay, so it doesn't affect the order of the amino acids or anything. Okay, it only affects the overall 3D structure. Okay, because we know that these when it basically breaks apart this individual molecule into its original primary structure. I'll show you here what I mean. Because we know that to make a full-on protein, we need amino acids that form polypeptides. They can be folded in weird ways to become a helix or a beta pleated sheet, and then these can interact with each other to form a full protein. So denaturing is breaking this 3D structure and reversing it back into pretty much the original chain of amino acids. That's what denaturing means. But again, if you don't exactly get what I mean here, you need to go check out my teaching video where I explain this much more slowly and carefully. This is just a run over of the questions. So is it A, the order of amino acids is changed when the protein overheats? So we know one of the things that can cause denaturation is temperature, right? So when you increase the temperature, you can denature a protein. Now, the thing that is wrong about A here is they're saying the order of amino acids can be changed. But I just emphasized that when you denature something, you pretty much return it back to its original structure, its original primary structure. You don't change the order of these amino acids. All you're doing is you're undoing all these foldings that happen. That's all you're doing, okay? So cannot be A because the order of amino acids is not changed. Now what about B? The bonds between amino acids are broken by condensation. First of all, there's two things wrong with this. Condensation, one main thing wrong with this. Condensation is the action of forming a bond. So they're saying the bonds between amino acids are broken by condensation. It should rather be hydrolysis. So that's what's wrong with B. What about C? Parts of the protein become linked together by hydrolysis. Again, this one is wrong because when you link things together, that's called condensation, not hydrolysis. When you break something, that's called hydrolysis. So B and C are kind of doing the same thing but the opposite. Okay, the word they're using to describe the process is wrong. So that means it has to be D. So let's check D. The three-dimensional structure of the protein is altered. So like I said, a protein can be 3D in shape with all the foldings and all these helixes and beta pleated sheet bonded together and forming this kind of 3D structure. Now denaturing breaks this 3D structure back into the original primary chain of amino acids. Okay, so that's true. And we know based on this here that a protein takes a specific shape for a specific function. So the 3D structure of the protein is important in determining the specific function it's going to have. If this 3D structure is broken, let me show you here. If this 3D structure is broken, now think about it, these spaces here where it was supposed to bind to something is also changed. And so maybe now by destroying this 3D structure, by denaturing a molecule, it will not be able to do its original function, okay? So that's why it's D. So the 3D structure is very important for the original function. Next one. The image here shows the structural formula of a molecule. Okay, so again, structural formula just means the actual way you draw out the molecule. So we can see here automatically that this is an amino acid because we can see the N. We can see the N, this amino group here. We can see it's bounded to a carbon, which is bounded to this R group. So this whole thing right here pretty much represents the R group. Okay, and this one here represents our carboxyl group. Let me show you. So see here, we have our and yeah, maybe you noticed something here. You saw that there's NH3 plus, and then suddenly there's a hydrogen missing here. Basically, this is more complicated than what you need to know as a biology student. But when you see this NH3 plus and hydrogen here missing, it's still an amino acid, okay? It's still an amino acid. All that happened was this hydrogen from here moved to this nitrogen here, but we still call it an amino acid. And again, don't worry about this. This is not something you're expected to know. Just know that it's still an amino acid in this case. So notice this here, this thing here. This is the R group. So remember we talked about an R group. I say that it can represent any sort of molecule. So this is any sort of molecule. This is an example, okay? Another molecule can be here as well, okay? So this is the R group. So this is obviously an amino acid. Okay, so I chose this question just because I want you to be aware that sometimes they will ask you a question that requires the knowledge from a bunch of things. So let me show you here. We know that in this chapter, chapter 2, we learn about lipids, protein, which is what this video is about, but carbohydrates and nucleic acids, right? So we learn about all of them. Now this is an example of a question that the answer requires you to know about all of these, okay? Not just one of them. So it's mixing different topics. So maybe you won't be able to answer fully this question, but I just want you to be aware that questions won't only be on one specific thing. Sometimes they mix and match these questions. Let me show you. So which types of molecules are shown in the diagram? So they show you 3 molecules. We can recognize that this is the amino acid, right? The amino group, the carbon, the R group. See, in this case, the R group is simply an H, a hydrogen. Whereas in this question, the R group was really complicated, okay? So, and then we have the carboxyl group. Molecule 1 here is a ribose because it has 5 carbons, 1, 2, 3, 4, 5. It's a type of sugar. And then this molecule right here is a type of lipid, okay? Because I'm not going to explain in this video, but you'll need to know this for topic 2 anyways. Now anyways, It's not important that you know how to answer this question, because this question video is just about proteins, but I just wanted you to see that they mix and match them. So molecule 1 here, as we know, is a ribose. Okay, so automatically we know it's C just by knowing that it's ribose. Now, can we confirm this? Let's check. Molecule 2 is an amino acid, and we can see that's true here, amino acid. Molecule 3 is a fatty acid. It's a lipid. Okay, so the answer will be C. Again, doesn't matter if you couldn't do this because you needed to know about the ribose and lipids and stuff, which is not part of this title of this video. So, but anyways, it's good to know that they mix and match, okay? So next one, the last one of this video, which type of molecule is shown in the diagram below? So this one is pretty complicated, right? You're like, wow, this is complicated. How on earth do I know this? But if you look carefully, you'll know exactly what this is. Notice here, if we start at the one edge, we can see this amino group, okay? And it's bound to a carbon, okay? And this right here is the R group, the R group. It can be at the top or at the bottom. Don't be confused, okay? It depends, you can draw it either way, as long as it's attached to this carbon here. So you can see the R group, and then we have this carbon, but we don't see the hydroxyl group. I mean, we don't see the carboxyl group, right? Because when you look at the amino acid here, if we look at one amino acid, there should be a carboxyl group. But remember, when two amino acids bind together, this carboxyl group will mix with the amino group of the next amino acid. Let me show you. So if we have two amino acids here, what will happen is basically the hydrogen from the amino group will interact with the hydroxyl group, or this OH group from the other amino acid, and it will leave as water, okay? So it will leave as water. You can see H2O, okay? There's two H's and an O. So it leaves as water. This is called a condensation reaction, and then the bond formed will now be between this nitrogen here and this carbon directly, because remember, each carbon wants to make 4 bonds, but if you remove this oxygen and hydrogen, now there's a bond that it has that it cannot make, okay? It's missing a bond. It would like to make this bond. It's basically like this. Think about it. It's basically, it has an open bond, and when the hydrogen leaves from the nitrogen is missing a bond. So they pretty much just decide to bind to each other. This way, the carbon has the 4 bonds it wants, 2 to oxygen, 1 to carbon, 1 to nitrogen, and the nitrogen has the 3 bonds that it normally wants, okay? So this is called a peptide bond. So you'll notice now when we look at it, we have this normal, we have the normal amino group on one end and the carboxyl group on the other end, and then instead we have this peptide bond here, which means that we can't clearly see a carboxyl group or amino group anymore, okay? Because there's a bond that happened. So when we look at this molecule, on the one end, we can see an amino group, and on the other end, we can see a carboxyl group. So automatically we should know this can indicate that multiple amino acids have been joined together. How many exactly? Let's see. NCC, that's one amino acid. NCC, that's another amino acid with a different R group. See, this R group was this amino acid has this kind of R group, and then this amino acid has this kind of R group. So we can see that overall there was 3 amino acids here binded together by peptide bonds, okay? So what we formed here by linking together multiple amino acids is a peptide. We call it a peptide, okay? So the answer is A. So this was honestly much more complicated of a question, because initially when you look at it, you might be just, wow, what is this? You might have no idea, but you just have to look very carefully and notice and look at what you learned, okay? They're never going to ask you something that you didn't know or shouldn't know about, okay? So just look carefully and analyze it properly. Okay, so that's it for this video. I will make another one, a part 2 on this, so stay tuned for that.